We're now ready to talk about esters as the functional group of acid derivatives, and it's probably going to take 3 lectures to finish this. Alright, so the 7A lecture that I'm going to spend next 10 minutes or so is about where esters are found in nature, because when we talked about the 4 derivatives, remember I said the acid chlorides are so reactive, it's really not found in nature. And hydrides are found to some extent, as you'll see in a bit. But esters and amides are a lot more stable than acid chlorides and anhydrides. So these 2 functional groups are found in nature everywhere, which is why you really need to understand the ester and amide chemistry, and we'll be using them again in later units, unit 5, when we talk about biochemical compounds. All right, so this lecture I want you to be able to recognize the ester functional group in all these compounds that we're going to be briefly going over, and then we'll go over the nomenclature and reactions in the next 2 lectures. All right. So in Unit 5, we'll come back again when we talk about fats and oils. There's a whole unit on biochemistry, and part of that we'll be talking about lipids. And fats and oils belong to that class. And if you look at the structure carefully, notice that there are 3 ester functional groups. See the one O here, one's here, another one, another one, right? And the reason why they are called triglycerides is because of the 3 ester group off of this glycerol, which is 3 carbon long with OH on each of the carbons. And that's why fats and oils are classified as triglycerides. So we'll be coming back to this, but basically the difference between saturated fat and unsaturated fat is whether or not the R group on the ester is saturated, as in this example here, all just carbons and hydrogens in single bond. This is what's found in animal fat usually. And the reason why they're solid at room temperature is because they're able to be stacked nicely, as you can see, to form a solid at room temperature, whereas the oil are unsaturated triglyceride, so their R group has kinks in them. They're all cis double bonds, and because of this kink in their chain, as you can see in the space filling model, it's very hard to stack them nicely compared to the saturated part, so therefore they exist as liquid at room temperature. All right. Now, because we're so self-conscious of our weight and things like that, and want to be healthy, chemists have come up with a fat-free fat called olestra, and there's a controversy about this compound. And the reason why I'm showing you this is because you don't need to draw it or anything. I just want you to recognize the structure. You should recognize this backbone. What is that? This backbone is a disaccharide, so it's a sugar molecule, and it's actually structure of sucrose, or the table sugar. So what they've done is, instead of taking glycerol as the alcohol to make the esters, they've taken all the OHs in sucrose and have put long chains on it as an ester group. So this is like the very, very, very big version of a normal fat molecule. So compared to normal, this is, you know, several times bigger with all these R groups coming off. Okay, and we'll come back to this olestra controversy when we talk about lipids, but I'm just showing you because of the structure. Now you should be able to recognize the carbohydrate plus the ester functional group to make it the fat-free fat. All right. So we'll come back to the triglycerides and cholesterol later on when we talk about fats. And the other places where you see esters is flavoring agents. So here's like a table of different esters, and we'll go over the name later on, but basically all these esters represent different flavor that is used to mimic banana or orange or apple or pineapple or grape flavors. All right, and so the flavoring agents here, for example, methyl butanoate is an apple flavor, ethyl butanoate is a pineapple, and etc., etc. So if you look at the apple versus pineapple, there's only one carbon difference because they're both of butanoic acid, and one is methyl versus ethyl R group. All right. So flavoring agents is used routinely to synthetically give you that flavor, and actually in lab you can make these very easily. And in the past we've had labs where you go and make, you know, banana flavor esters, for example. All right. The other type where esters are found are pheromones, which are ways that insects and animals communicate with each other by these chemicals. This right here is an ester, as you could see, and is a sexual attractant that's used by female dogs when they're in heat. That's what they excrete, and which is detected by male dogs. And then this also is it's a type of pheromone because this is the active ingredient in catnip. And notice that this is a cyclic ester. Okay, now cyclic esters have a special name called lactone, and the lactones are found in nature everywhere as well. So I just want to basically, I'll give you an example so that I could tell you what these cyclic esters are called. All right. Now, in medications, we find ester groups everywhere as well. This is the structure of salicylic acid. And if you take this alcohol and then make it into a methyl, into acetic acid ester, then what you're creating is aspirin. Whereas if you functionalize this carboxylic acid and then make the methyl ester here, this is oil of wintergreen. And this oil is often used to decrease pain on sore muscles by rubbing it on. So these are some of the just examples of esters found in nature. All right. Now, esters are also found in man-made polymers called polyesters, and these are some of the examples of polyester. All right. So I'm sure Dr. Gurney talked to you about polymerization and what polymer is. And you've seen it with sugars, you know, when you have a monomer and then you link it together and you have hundreds of these units together, you make a polymer, right? So in this polyester, we have two units coming together. So notice the one unit has acid on both sides, and the second monomer has alcohol on both sides, because that's how you put together an ester functional group. So think of each, I'm going to use my son's Lego. So think of this diacid as a red Lego, and think of this diol as a green Lego. And when they come together and form this bond right here, so the bonds that are forming is here and here. And it keeps going because they're functionalized on both sides. So you make an ester linkage here, and then you put another acid here, right? And then another alcohol, So in other words, you could kind of connect them together in a AB fashion like this. So think of this monomer as A, think of this as B, and then you have AB monomer, polymer, that keeps repeating AB like that. And then because the linkage is ester, this is polyester, and you could have thousands of units like this. And you've heard of polyester type of polymers that you in clothes, for example. This is an example of polymer that's derived from glycolic acid with lactic acid. And notice in this one you have a carboxylic acid and an alcohol, both functional groups in one, but you have an alcohol carboxylic acid as well. So you're still doing the AB type of polymerization. And this is actually a polyester that is used in stitches that you get when you go to emergency room, for example, and they sew you up. And this is a type of thread that you don't have to go and take it out, because over time it slowly hydrolyzes. So the ester bond is cleaved slowly automatically, so that it just sort of goes away. All right. The reason why I put this polyester is a polylactic acid. Lactic acid right here has a carboxylic acid and an OH. So if you make a polymer AA like that, same unit repeating over and over again, you get a polylactic acid, PLA cups, which is what NatureWorks produces, and that comes from corn. And the polylactic acid polymer it's great because it depolymerizes readily, and what you get back is a lactic acid, which is not harmful to the environment. Okay, so we spent actually 3-4 years of our research time studying this depolymerization reaction, and then actually using lactic acid, because it's chiral in an asymmetric reaction. All right, so I expect you to be able to draw, given the monomers, be able to draw polymers after you learn about the ester chemistry. And then if I give you a structure of polymer, be able to recognize what a monomer would be that it came from. All right. Now, esters and anhydrides are also found in inorganic portion as well. So I'm just throwing this out to you for recognition. You've heard of ADP, ATP, and AMP. This is the structure of it, and you recognize the sugar, and we saw this part. Okay, now if you put the phosphate groups on, notice this is really an inorganic ester, okay, POO, but then here is really inorganic anhydride, right? So I just want you to be able to recognize that it doesn't have to be just carbons in order to be esters and anhydrides. You could have inorganic stuff as well, okay? And then this is an alpha D glucose 1-phosphate, so here's another inorganic ester as well. So I'm going to stop here and finish in the next lecture, so I don't run out of time.